T-cell development takes place within the thymus, a specialized organ where T-cell progenitors mature and differentiate. The thymus is divided into several different anatomical locations, and as thymocytes mature, they pass from one environment to another. T-cell progenitors enter the thymus through high endothelial venules and migrate to the subcapsular region, where these thymocytes start their process of maturation by rearrangement of their T-cell receptor genes. As maturation proceeds and the cells begin to express a specific receptor, they move further into the thymic cortex. Interactions between the T-cell receptor and MHC peptide complexes displayed by the thymic cortical epithelial cells play an important part in the fate of the thymocyte. At this point, the developing thymocytes express both CD4 and CD8, as well as an alpha-beta T-cell receptor. A cell that has a receptor able to recognize an MHC class I molecule receives both a survival signal and a maturation signal. Eventually, the cell stops expressing CD4 and maintains expression of CD8. Thymocytes that obtain a survival signal are said to undergo positive selection. A cell that has a receptor able to recognize an MHC class II molecule also receives a survival signal and a different maturation signal. Eventually, this cell stops expressing CD8 and maintains expression of CD4. Thymocytes whose receptors are unable to recognize either MHC class I or MHC class II molecules fail to receive any survival signals and die by programmed cell death, or apoptosis. Thymocytes that are able to recognize MHC class I or MHC class II peptide complexes too avidly receive a strong signal that drives them into cell death. In this way, thymocytes capable of responding to self-peptide antigens are eliminated in a process known as negative selection or central tolerance. The surviving T cells migrate from the thymic cortex to the medulla. At this stage, they remain capable of recognizing self-peptide antigens expressed on other cell types, such as dendritic cells or thymic macrophages, and receive a sufficient signal to cause them to undergo programmed cell death. The remaining thymocytes, now mature naive CD4 and CD8 T cells, pass out of the thymus, either returning to the bloodstream directly, passing into venules, or via the lymphatic system.